How can we fight one of the greatest wars known to man, the war against cancer? T cells, our body's fighter immune cells, are born to defeat pathogens, but often, in the case of cancer, they're deactivated. My name is Sidharth Raghavan, and you're listening to Entangled Strands, a biotech podcast that seeks to inspire budding scientists and delve into the mysteries that lie within us. In this episode, I'll explore how we can reprogram T cells to fight against cancer, and I had the great privilege of talking with Dr. Maksim Mamonkin, an assistant professor at the Baylor College of Medicine. Dr. Mamonkin's research focuses on the immunobiology of CAR T cells, and he is using protein and gene engineering techniques to target malignant T cells, such as T-cell ALL. His new therapies are now being tested in phase 1 human clinical trials at the Baylor College of Medicine. For those who don't know, CAR T-cell therapy works by extracting a patient's fighter immune cells, their T cells, and reprogramming them by inserting a CAR gene that helps them recognize cancer cells. Our conversation begins by discussing autologous CAR T-cell models and problems with cell fratricide, cells killing their brother cells. We then discuss allogeneic CAR T-cell methods and what lies in the future for CAR T-cell therapy. You can learn more about Dr. Mamonkin and CAR T-cell therapies by reading this episode's notes on brooklynmusings.com. And you can view previous Entangled Strands episodes on Spotify, iTunes, YouTube, or directly on the Brooklyn Musings website. Enjoy the podcast. So I first wanted to ask you how you became interested in science. So what was that inspiration like for you?
Well, it's an interesting question. I really don't know the answer. As long as I remember myself, I was interested in learning something new, right? And science is all about that, learning and kind of testing your hypothesis. So I think it's just a natural trajectory for me. And as for many others as well, I was exploring medicine initially, but then realized it's probably not a good fit for my mind or my brain. I don't know how you say, but it's been a journey that I've thoroughly enjoyed so far. So
that's great. So what was your college life like? What did you study in college? I read that you did college in Russia at Novosibirsk University. So what was that like for you?
It was great. I must say the higher education in Russia is pretty good, and it is free for most. And so a lot of people take advantage of that. As in, we go to colleges and study what we want, with little regard of how to pay for this. Unlike things are here in stateside. But initially I was very interested in chemistry from high school. I was participating in all this competitions and Olympiads and like national, international level. But I soon realized that it's becoming too boring for some reason, and I kind of became interested in biology, which to me back then, and pretty much still now, kind of seemed like chemistry taken into just a new level. So we figure out the chemistry of the cell, how a cell works. And more so in concepts rather than the actual chemical reactions. And so that kind of sparked my interest in biology. And so I studied biology, microbiology in particular, in Russia. And the system that we had there was kind of a hybrid bachelor master thesis. We study 5 years in total. In the last 2 years, we spent a lot of time in the lab. Working on a research project. And so that actually exposed me to a lot of experimental things. And I was studying inhibitor receptors on NK and T cells, which further kind of bolstered my interest in immunology. And then I kind of realized that I really want to do it for the rest of my life. And that's how I ended up here stateside in Houston. I joined Baylor for the graduate school in immunology and never looked back since.
That's great. So, one thing that I noticed is that you did your PhD at Baylor College of Medicine. And how is that different from, let's say, doing a PhD at a biology department at some other university? How is this experience different?
Well, I can't really compare. I did my PhD at Baylor, as you said,
Right.
and here, I think graduate programs across the schools are quite similar in terms of the curriculum and the format. As we take care of our curriculum courses in the first year or two, and then pass a qualifying exam, and then focus pretty much the rest of the graduate school and doing thesis in the lab. And so I'm not sure if the experience, let's say, at a college of medicine is different from the experience at a large university, with probably an exception that we don't have a lot of undergrads here. So as a graduate student, I didn't have to teach a lot. Although I did have to still participate in some teaching to medical students and to graduate students, and I still do that as faculty now, but we don't have undergrads. So that's the kind of the difference. But all in all, I think it should be similar to other schools.
Got it. So you were already interested in immunology from your undergrad times with looking at NK cells. How did you get interested into CAR T cells and adoptive T cell therapies?
Right. So my PhD was not in CAR T cells. It was actually in fundamental immunology and T cell immunobiology, where I became very interested in, as I said, studying how a cell works. And T and NK cells were fascinating to me just because they have unique function, and the way they work and the way they're regulated, particularly how their differentiation is governed, what determines whether they become memory cell or an effector cell is a big enigma, and I think still is in the field. We still don't have a very good understanding of the factors that drive the decision to become a memory cell versus a short-lived effector cell. And that's something that drew me in the field. And I was studying transcriptional regulation of CD8 T cell differentiation in mouse models using Listeria infection. And so basically looking at the effect of particular transcription factor knockouts in mouse models to see how that shapes T cell differentiation. And that's really exposed me to a wealth of knowledge in the field, in the T cell field, but also to cutting edge experimental models, which is something that I was always interested in. And one thing that I realized probably later, closer to the end of my PhD, is I don't have a single textbook in immunology. Because I studied immunology not by textbooks, but just reading papers, and because you can learn so much more from just reading papers, discussing them in journal clubs, than you do by just reading a textbook. So that's how I learned it, and so at the end of the grad school, my project involved studying a couple of transcription factors, one of them KLF4, cryptolite factor 4. And another is a NETs family factor. And we also, I had like a short collaboration with one of the faculty here in the Center of Cell and Gene Therapy, studying the effect of this gene knockout in the context of human T cells. Like, what will happen if we silence it? Knockouts did not exist back then. We didn't know how to use them. It was 2011. And so we silenced it, and to my dismay, nothing really changed. Like in T cells, they didn't even budge, they didn't even notice that. And I was so upset because it had such a huge phenotype in mouse T cells, but barely anything in human T cells. And there's a lot of mouse genes that behave very similar in human T cells. I guess I didn't get lucky in picking the right gene. And so I kind of decided after the grad school that I want to continue studying something that matters in human T cells. And the best way to do it is actually to switch studying human T cells. And what drew my attention, interest in CAR T cell and Doppler cell therapy is not the CARs themselves. A lot of people like me with the training in fundamental immunology were kind of agnostic of CARs. It's a fraud. It's just something that it's a fad and it will just go away. But I was mostly attracted to engineering part of it, like messing and tinkering with signaling pathways inside T cells and make T cells do whatever we want them to do. And CARs could be just a tool for that, but I was more attracted to the possibility of bioengineering inside the T cells rather than just, well, let's make a new CAR and see how it works. And so that's how I ended up in the center as a postdoc with Malcolm Brenner.
So then you moved on to, you developed some CAR T cells that can be used against T cell cancers, right? So there's the CD5 version and also the CD7 version. And so before we get into that, what already exists are these CAR T cell treatments for B cell cancers. So there's Yescarta, Chimera, and a couple others. And how is treating B cell tumors different than T cell cancers? Because it seems like it's harder.
Oh yes, it is absolutely a different game. When I started, it was late 2013, early 2014. There was no Kymriah or Yescarta. There were early initial results that were very encouraging, CD19 CAR T cells and B cell tumors. And actually, as I mentioned before, I was primarily interested in not just creating another CAR, but rather, looking at how to make T cells work in the modality that we want them to work. And so one of the projects that I had on the side is sort of to develop a system that was based on the CD19 CAR that would allow CAR T cells to more specifically target malignant cells and spare normal B cells. And I started looking at CLL, chronic lymphocytic leukemia, which is defined as a sort of a that's mature B cell malignancy that expresses CD19 and CD5. And so I thought, well, what if we create a kind of a dual system of receptors that would allow T cell to recognize a CLL cell that expresses 19 and 5, which is very unusual for B cells, to co-express the two. Some B cells do express them, but they're a minority. And so if we could create such a system of receptors that would specifically recognize CLL and will spare the vast majority of the B cells, then we can just use that as a safer alternative to just mono-targeting CD19 CARs. And so just to kind of make sure that the binder, so I kind of looked at what are the options for the CD5 binders, and I found an old CD5 specific antibody on NCBI websites that was used back in the early 90s in clinical trials in T-cell malignancies. And I kind of constructed a single challenge variable fragment based on just this kind of sequences of the antibody. And I didn't know if the single chain format would recognize CD5 or not. So I made a cytotoxic receptor and thought if T cells kill each other, then probably works. And that's kind of how it all started. And to my surprise, I did see some evidence of T cell targeting each other once I expressed the CD5 CAR, but then eventually it took off and started growing like crazy. And so kind of that precipitated the switch from the dual receptor system into like, well, this looks interesting. Let's try to develop this further as an interesting CAR that evades cell targeting through an interesting mechanism. And so that's how it all started eventually.
Wow, that's great, because that's what science is all about. You just get on a certain track and then you follow that track and see where it takes you. So you talked about CD5 and CD19. So you identified that these were proteins that are on these CLL cells, and then you engineered these CAR T cells to target both of those proteins. So hopefully it would just target those proteins which are on these CLL cells. Is that right?
Yes, exactly. So the idea is to kind of engineer this Boolean AND logic, right? That a
Right.
pair of receptors will recognize CD19 and CD5, and only then, if a cell co-expresses both of these proteins, then the T cell would degranulate and kill the target. But if it only expresses either CD19 or CD5, which would be the vast majority of normal cells, then the act of killing will not happen, just because it won't comply with that Boolean AND gate. So yeah, that was the original idea.
And that's really cool to think that a cell could exist where it's very specific in its targets. And yeah, I just find that very interesting. So you have these CD5 cells for treating T cell tumors. How do those work, and how are these T cell tumors different than the B cell ones?
Right. So I just realized I didn't answer your original question.
Yeah.
So this the whole premise of targeting T cell malignancies by normal T cells have to kind of we have to think about how to separate the on tumor activity from fratricidal activity, because the vast majority of the antigens, including CD5, that are expressed on malignant cells, they're also expressed on normal T cells. In fact, more than 99%, and maybe it's 100% of all T cells express CD5. And so, how do we engineer a solution that would allow us to redirect normal T cells, good T cells, against bad T cells? And so this problem doesn't exist in B cell malignancies, because the antigens that we're targeting there, CD19, 20, 22, BCMA, they're not expressed at high levels on T cells at all. So that really doesn't trigger any noticeable fratricide. And fratricide is the kind of self-killing or self-targeting of T cells. And so that problem has to be solved just to allow the expansion of CAR T cells, right? And so we can target pan-T cell antigens like CD5, CD7, CD2, CD3, but we have to do something to avoid T cell killing each other, because the vast majority of them also express this receptor. That's one problem. Another problem is when we infuse CD19 CAR T cells to patients with B cell malignancies, they do not discriminate between malignant B cells and normal B cells, and so as a result, normal B cells are also ablated for variable duration of time, and so that luckily can be managed by supplementing with IVIG and kind of offsetting that risk of immunodeficiency. But similar approach will not work for T cells, because T cells cannot be supplemented with something soluble. They're a component of cellular immunity, so they have to be. If they're ablated, we all know what happens. And so this is a much more dangerous situation in the clinic that we have to worry about. And so these are the main problems that exist in T cell malignancies, but really don't exist in B cell tumors and other malignancies as well. And so that presents a very interesting bioengineering challenge, right? How do you overcome these limitations and engineer therapy that is effective and safe in these patients? And so, we started that journey back in 2014, and we're still learning something new every day.
Wow. So you talked about self-fratricide and how that's a major problem. So what are some of the ways that we can stop the self-fratricide from happening?
Right. So there are different mechanisms that either exist in the nature that protect normal T cells from fratricide. Because remember, T cells themselves, they express these cytolytic granules that are designed to kill other cells. And so they have developed mechanisms that protect themselves from their own explosive cargo. They have genes that resist the activity of granzymes, that inhibit the activity of perforins. And that really allows them to throw these grenades without being, hit by themselves, by these grenades. And so those are kind of inbuilt mechanisms, but they are often insufficient when we express a powerful receptor on the cell surface, like a CAR or even TCRs that target antigens that are also expressed on T cells, such as survivin, for example. And those studies were done way before we started our CD5 project. So we knew that T cell fratricide is a problem with engineered receptors. But it just so happens that in the case of CD5, T cells actually avoid fratricide automatically by very rapidly down modulating and internalizing and degrading CD5 protein after the CAR is expressed. So what happens is once the CAR shows up on the cell surface, CD5 is internalized and then eventually broken down. So we have my graduate student currently is finalizing this story. It's a very, very efficient process that happens in all T cells once we express that CD5 CAR. And at the end of the day, the entire protein, CD5 protein, is eliminated. We don't see it by flow cytometry. We don't see it by Western blot. It's gone. But the CAR is still there. And so that allows these T cells to still kill CD5 positive targets. But by themselves, they are kind of invisible to each other because they degrade their target protein in themselves. And so that is a property that we think is more or less unique to CD5. We really don't see the same thing happening when we target other antigens that are highly expressed on T cells. I'm not saying that this is purely unique to CD5. There may be some antigens that behave the same way, but we just haven't really seen this with anything that we have developed here in the lab. So that's one mechanism that can help avoid fratricide. And as I said, it's probably kind of more or less unique to CD5. Now, if you target other antigens that are highly expressed in T cells, such as CD2, CD7, even CD38, that gets upregulated, there we start seeing very robust self-targeting of T cells. And so there we can figure out a way to engineer this resistance, right? We don't have to engineer anything in CD5s, but we do have to engineer it with other targets. And so one approach that we've developed back in 2016, 17 is just to kind of edit out the target antigen. So basically you delete its or disrupt its expression in the genome using genome editing tool. So we use CRISPR as an example. So we took CD7 CAR T cells. Well, we actually took T cells, stimulated them, that we knocked out the CD7 gene, and then expressed the CAR in there, and that allowed the T cells to expand very nicely. And we had that published also back in 2017 with my first graduate student who I developed these CAR T cells for both T-ALL and AML. About a quarter of AMLs express CD7 as well. So he really kind of, with the help of others, helped kind of establish this technique in the lab. But originally, right now CRISPR is widespread. We can CRISPR out anything anywhere. But back then, it was just emerging. And so we got very lucky that another lab in our center, Peggy Goodell, they had a couple of fellows there, Mike Gundry and Lorenzo Bernetti, who kind of figured out and optimized a way to CRISPR out genes in hematopoietic cells, stem cells initially, and then another good friend of mine from Germany, Dimi Wagner, he was an exchange student in our lab. He was fascinated and adapted that approach for T cells. And we were chatting over beers one night. And so we made the CD7 CAR and they're just killing everything. He was like, why don't you just knock it out? When in doubt, knock it out. And I'm like, you're out of your mind, right? So that's how it all started. And so we showed that this is a very effective way to avoid fratricide. And pretty much the same year, there were two other reports from other groups that also looked at the ways to prevent fratricide in CD7 CAR T cells. One was from Washington University, John DiPersio, but they actually took one step further and they knocked out both CD7 and the CD3 genes to create an off-the-shelf version of CD7 CAR T cells. Their kind of platform was a bit more advanced than ours. And then another group was Daria Campana's group in Singapore, who explored a non-gene editing approach disrupt CD7 expression, and they achieved that by essentially trapping the CD7 expression inside the cell using what they call pebbles, or protein expression blockers, which were kind of the short CD7 specific binders that were anchored in the ER through the KDEL sequence. And so essentially they bound CD7 and retained it inside the cell, didn't let it kind of appear on the cell surface. And that was enough to hide the expression from the neighboring cells and prevent fratricide. So this is an alternative way, very clever way, I should say, to avoid the fratricide. But all in all, the conclusion was that you have to do something to disrupt the cell expression. Otherwise, the fratricide is just very, very strong.
So why is CD7 expression on a CD7 CAR T cell, why will that result in cell fratricide? So why do you have to stop expression, knock it out, or do something with that to stop the cell from attacking other CD7 T cells?
Right. So, as we touched on earlier, the CAR T cells in their current iteration that really don't discriminate between, malignant cells and normal cells that express the same antigen. If they see the antigen, they will kill the cell. And so all T cells express very high levels of CD7. Well, not all, I should say, about 85 to 95%, depending on the donor. Most of them express CD7. There is a population of naturally CD7 negative cells, both CD4s and CD8s. And those would be resistant to fratricide just because they don't express CD7. But the vast majority of T cells do express CD7. And so one way that actually is being developed right now by Julian Velasquez and Steve Gottschalk and St. Jude's is to actually enrich for those naturally CD7 negative cells up front and then transduce them with a CD7 CAR and then expand. And so you don't have to edit out anything because the cells are naturally CD7 negative, right? So they won't see each other. And we have developed recently an alternative approach where we transduce T cells with CD7 CAR and then expand them in the presence of pharmacologic blockers of CD7 signaling. And that kind of allows them to grow, but the CAR signaling is suppressed, that there's no fratricide. But once we infuse them to the mice, they undergo fratricide there, but they also killing target cells. And then in the end the natural CD7 negative population kind of takes off and survives this whole thing and establishes kind of long-term persistence. So that's sort of a natural selection of those cells that's happening in vivo. But the bottom line is, you have to, just because the vast majority of T cells express CD7, they would be targeting themselves, but you could, either disrupt their expression forcefully, or you could select a population that is naturally fratricide resistant because they don't express CD7 mRNA. So all these approaches make sense, and they've been shown in preclinical models to be effective. And the question is which of them, or there may not be just one. I mean, all of them may be effective in the clinic, right? But they're all very different, and so we'll see how that pans out. We actually have one, the clinical trial based on this naturally selecting as natural selection approach right now, open at Baylor, and we just started enrolling patients, so we'll see how it goes.
That's great. So are there any other clinical trials that are going on with these CD5, CD7 CAR T cells?
Yeah, so the CD5 trial, we've actually opened it back in 2017 or 2018, I don't remember, it was a while ago. And so this was the first in man CD5 specific trial, and just because we weren't sure what the safety of this was, what would be the effect of normal T cells that were overwhelmingly CD5 positive, we designed it as a bridge to transplant, which means that patients with refractory relapsed disease that does not respond to any other treatments, they would receive infusions of autologous CD5 CAR T cells. And then if they respond to that treatment, that would proceed to transplant, which would kind of terminate the activity of CAR T cells if there was any unwanted activity against normal T cells, and then allow them to proceed to transplant and kind of reset the hematopoietic system to allow T cell restoration. And so it's been going for several years now. We are approaching the end of it, and we actually got very promising results in both T cell lymphoma and T cell ALL patients with leukemia. And we're seeing very nice activity and pretty good safety profile of the cells. And that's another interesting peculiarity of CD5 CAR T cells is they can expand in patients and persist and eradicate the malignancy and produce complete remissions. But even in those patients where the tumor completely kind of melts away, we're still seeing normal T cells that survive the activity of CD5 CAR T cells. And this, again, this is a very interesting mechanism that allows them to escape this activity by transiently downregulating CD5 in trans in close contact with CD5 CAR T cells because of those mechanisms that exist in normal T cells that protect them from their own cytolytic machinery, right? And so there's a population that is spared, and they remain in circulation, protect patients from complete T cell aplasia. So this is a very interesting finding that we saw in these patients. But all in all, we were able to kind of continuously improve the GMP manufacturing process of CD5 CAR T cells during this study, and eventually we were able to kind of determine the factors that affect the activity, sustained activity and persistence of CD5 CAR T cells. And now we're seeing pretty good responses in both T-ALL and T cell lymphoma patients, with a lot of complete remissions in patients who really don't respond to conventional treatments anymore. So we are very excited about that, and at some point the phase 1 will conclude, and we'll see where to take it next.
That's great, and it's also very interesting, as you said, to see these CD5 T cells being able to, on their own, stop themselves from attacking their brother T cells, which is great. So we talked about this is an autologous CAR T cell method where you're taking the patient's own T cells and you're re-engineering them. So what are the problems with having a method like this when you want to expand it to a greater population? And what are the problems with that and current T cell therapies, CAR T cell therapies?
Well, I mean, I guess the problem you're alluding to is the fact that we have to manufacture these cell products for each patient individually, right? Which takes time. The final product is quite heterogeneous. We can't really ensure its potency. We just manufacture to see if we can grow the cells, and which sometimes is a problem with some patients, as particularly after chemotherapy, several rounds of chemotherapy that produces a lot of damage to circulating T cells and other lymphocytes. And so, the fitness and frequency of T cells in the blood of these patients is quite low. And when you sort of expand them in a T cell product, their potency is also quite limited, because during the process of expansion, they further lose this capacity to persist. The more they proliferate in the dish, the less they proliferate in the patient. And so, we're seeing this firsthand, that, in most patients, it's still enough to produce good therapeutic benefit. But there are patients who are riddled with a disease, and has such florid disease that we can't really manufacture a cell product just because there's so many circulating malignant blasts in the blood. And another big problem of autologous therapy, particularly in patients with very aggressive diseases, is that they progress so quickly between the time that they're kind of enrolled on the study and we'll collect the product. From the time we're ready to infuse, they progress so much that they become ineligible for infusion. And it's not just T cell malignancies; it's other leukemias as well, IML, and there's a lot of other examples. And so, obviously, one way to overcome these limitations is to have a therapeutic product pre-made and banked. And so it is available sort of off the shelf for anyone who has disease with a suitable phenotype. And so this would expedite the treatment. It will democratize the treatment, also decrease its cost. We can also ensure that the final product is very potent because we can test it beforehand and select good donors and right modifications to increase its potency. And so that would solve a lot of these problems with autologous therapies. Now, it also introduces two new problems that don't exist in the autologous setting, and those obviously have to be solved for these therapies to be effective and safe. And both of these problems are due to the fact that you're injecting someone else's cells into the patient.
So there are a lot of different ways that scientists have proposed making these off-the-shelf CAR T cells, like with any other treatment for cancer immunotherapy. And one of the innovations that I think is quite amazing is that your team has engineered this alloimmune defense receptor system that targets activated T cells and NK cells that usually suppress the immune system and reject these off-the-shelf CAR T cells. But if you can target those and you can kill those, maybe these off-the-shelf CAR T cells can survive in the body. So how did this method come about, and how does this work?
Well, I mean, first of all, I just wanted to make it very clear that we did not invent a new class of receptors.
Right, yes.
These are essentially your CARs, right? So you can call it a formally specific CAR. But we gave it a different name because it has a slightly different function than the conventional CAR. It is designed to be co-expressed with the actual CAR. And so just to not confuse it with another CAR, we give it a different name. But essentially, it's the same cytotoxic receptor that everyone is using. So, the main problem with, I think this becomes, actually the main problem in the allogeneic setting with off-the-shelf third-party donor-derived mismatched T cells is their immune rejection. You can make them, safe by editing out TCR so they don't produce graft-versus-host disease, or you can manufacture them from naturally non-reactive T cells or NK cells, right? So they don't have host reactivity. But preventing host rejection is a much harder task just because the immune system has so many ways to recognize alien things or things that don't look right. And that's part of the reason why we still exist on the face of the earth, is because our immune system is so versatile. It can detect just minor variations in cells and effectively protect us not only from infections but also from cancer and other things. So it's a very difficult task to engineer a cell product that will be completely invisible to the immune system. I'm not saying it is impossible. And it probably does not have to be completely invisible to the immune system, but it has to be invisible enough to persist and produce therapeutic benefit. And so, as you mentioned, there are other approaches that have been designed to address this problem. And, you could selectively inhibit NK cell activity by expressing, inhibitor receptors. You can knock out class 1 and class 2 to hide them from T cells, which is a very effective way to prevent T cell rejection. But it would also amplify the NK cell rejection. And, dealing with NK cell rejection is probably a little harder than most people think, just because NK cells are so heterogeneous and they express various sets of activating and inhibitor receptors. And so one receptor probably won't do the job. And it's possible to combine several of them, but this makes engineering task a little bit more challenging. And so, I'm not saying that those are inefficient. They probably will be efficient in the clinic, and we'll see that evidence soon. But we kind of decided to take on an alternative approach and kind of build on our expertise in targeting T cell antigens in the malignant setting. So we kind of asked the question, what if we extend the utility of CARs beyond the malignant space and actually redirect them to target pathogenic T cells rather than malignant T cells? And so a very one of the approaches that is currently very popular in the setting is to kind of fully ablate lymphocytes, like cellular immune system, by using CD52 antibodies, CAMPATH, and their analogs. And so that would kind of do the carpet bombing of the immune system, right? And just kind of eliminate all these lymphocytes that may reject the host. And so this is effective, and it prolongs the activity of infused cells, but it's also quite toxic just because you're ablating all T cells and NK cells, and that kind of predisposes patients to infections. And so we thought that, what if we make it a little bit more specific and selectively target activated cytotoxic cells, and spare, non-cytotoxic cells such as CD4 cells that really, they may play a role in immune rejection, but those are not considered to be the cells that directly reject the host. They amplify the response, but they're not very cytotoxic by themselves. And so we thought, what if we pick a surface marker that is specifically expressed on activated T cells that recognize and reject these allogeneic cell therapies? And so one of the antigens, surface receptors, that is very highly expressed on activated cytotoxic CD8 T cells and NK cells is 4-1BB, CD137. And so we thought, what if we just kind of selectively ablate 4-1BB positive cells by creating a 4-1BB specific CAR or a cytotoxic receptor? And so that's exactly what we did. It was a work of my second graduate student. So she developed actually a panel of receptors that target the main activation markers in activated T cells, including 4-1BB, OX40, ICOS. And though they all kind of pop up on activated cells and have very low expression on non-activated naive cells. And the idea was just to selectively ablate those activated cells and spare all the cells that don't really have anything to do with immune recognition, such as naive cells and CD4s. And so that's how it all started. And she eventually developed a receptor that was quite potent, and it was able to recognize those activated cells and protect T cells from being rejected initially in mixed lymphocyte reaction systems in vitro, against T cells, against NK cells, and then further she took it to in vivo and established an interesting in vivo model where the donor-derived cells were rejected in mice that were pre-grafted with T cells from another donor. And she showed that expression of this receptor is very effective. And so we called it alloimmune defense receptor, which just to kind of separate its function from a cytotoxic CAR that is often co-expressed with these ADRs. And we've shown that T cells that co-express the actual CAR, like a CD19 CAR or GD2 CAR, with this alloimmune defense receptor, they are capable of sustaining long-term anti-tumor activity in mice and resist rejection by alloreactive T cells in vivo. And so this is an alternative approach. I'm not saying this is better or worse. It's just another idea that we have at our disposal, and it can be combined with other approaches such as MHC knockout or expression of HLA-E or CD47 and other things.
Right. So there are a lot of approaches, including this one, that bank on, okay, we're going to make these T cells invisible from the immune system, or we're going to, I guess in terms of this method, we're going to eliminate activated T and NK cells. And so what kind of long-term ramifications could something like this have, where you're essentially eliminating very important parts of the immune system that, well, right now, if for a patient with cancer, it won't really matter, but maybe in 2 years when their cancer is gone, these cells may persist or may not. What do you think about that?
Yeah. So this is obviously a very important question. And again, it goes back to that theme of the that's been recurrent throughout our talk is that normal T cells don't discriminate.
Right.
As long as the target cell expresses the target antigen, it will be targeted by the T cells. And so, yes, it is a concern that obviously the 4-1BB specific T cells could also damage productive T cell responses, antiviral responses, maybe anti-tumor responses as well. And so we cannot really fully ensure against that. Now, there are reasons that made me think that these activity would be limited in the patients. First of all, not all T cells upregulate 4-1BB. Even in CD8 T cells, there's a population of cells that we see that don't really seem to upregulate 4-1BB upon activation. We don't really know why, what those cells are exactly, but we're seeing that not completely all T cells are eliminated. CD4s, the vast majority of them are spared just because most of them don't express 4-1BB upon activation. The second reason, which you kind of alluded to, is the persistence of the cells. And in an autologous setting, the persistence of T cells is pivotal because it's critical for the response most of the time, because we only have one shot at it, right? We have an autologous product, and if we inject it, that's it, right? It's difficult to remanufacture, or it is complex, and the patient's already past that point of eligibility and whatnot. And so the persistence of autologous cells is absolutely critical. When we talk about off-the-shelf cells that are banked and in theory available ad libitum, you really don't need that long-term persistence to achieve the desired effect. You can have a product that would persist enough to deliver a significant blow to the tumor, and then it would dissipate, and then you will reinfuse, and it will clean up the remaining cell, and you continue infusing them until the tumor is gone for good. But you don't want your off-the-shelf donor-derived T cells to persist there forever, just because it's not good for the cells, because they will go after tumor antigens that also may be expressed in normal T cells or normal cells. So even CD19, right? You don't really want your patients to be completely B-cell ablated for years, on end. And so as we start thinking about this modality of off-the-shelf cells, I think the persistence of the product becomes actually less important. It is important to have sufficient persistence, expansion, persistence area under the curve to ensure, decent anti-tumor activity in the first, let's say, 2-3 weeks, right? But you don't have to have that product persist for years and years.
Right.
And so, I think that's the reason why we designed this ADR aluminum deficiency receptor as a first-generation CAR, sort of first-generation backbone, without really the co-stimulation inside the cell, with the goal that the main persistence of the product will be dictated by the CAR and not by the ADR. And so, we think that if we engineer an off-the-shelf products in a way that it will not persist long-term, this activity will be limited, obviously. Now, there's a third reason, and that comes from our own experience in the center with CARs that target pan-T cell antigens, or antigens that are expressed very highly on activated T cells. One example is CD5. In theory, CD5 CAR T cells were supposed to ablate the entire circulating T cell compartment,
right.
But they don't, because of the mechanisms that we didn't know existed, right? Another example is CD30 specific CARs. CD30 is an antigen that's highly expressed in Hodgkin lymphoma and some non-Hodgkin lymphomas as well, such as T cell lymphomas. And we have ample experience in our center. That CAR was developed by Pietro Dotti, who's now at the University of North Carolina, and they're having a trial there. And they've treated many patients with Hodgkin's disease, and they really don't see a lot of toxicity in terms of T cell immunosuppression and whatnot. And CD30 is very highly expressed on pretty much all activated T cells. And that kind of tells you that the damage to the immune system may be limited. And keep in mind that in order for the CAR T cells or ADR T cells to permanently damage the immune system, they have to be in the close proximity to, like, in the same tissue, in the same compartment, with cells that respond to infection. And oftentimes the tumor and infection happen in different tissues, different parts of the body. And so, yes, there's some systemic circulation, but how much of that is actually going to be causing damage? We just don't know. The bottom line is we need to see how things pan out in the clinic. Yes, it is a concern, and we have access to all the mitigation strategies that, can help prevent long-term damage. But until we see how this works in patients, I'd be reluctant to table it just because of this concern. So we just received a large grant from NIH to move this forward to the clinic. So we'll be working on this. We're excited to kind of push this through clinical translation, see how things pan out.
Yeah, that's great. And that's one of the things that I didn't know about, which is that, a lot of these T-cell therapies, they don't really persist in the body for that long. And that's really great because, well, I guess we'll see from the clinical trials what exactly happens because you never really know. But so that's really promising, I guess, to at least know that it's worked in other scenarios. So
just one point that I wanted to make, just kind of to drive it home. So we tend to think about CAR T cells as living drugs, right? We inject them once and they self-propagate, they proliferate, they establish persistence, and they kind of live their own lives there. And it's great for when we target antigens like CD19, where we can afford, off-tumor toxicity that is on target. But when we start targeting antigens that don't have this favorable expression like CD19, if they express on critical tissues, such as HER2, maybe G2, or even CD5 or CD7, right? You don't want these cells to produce continuous ablation of these cells. And so when we start moving toward the allogeneic cells, I think it's very important that we start thinking about these cell therapies not as living, well, they are still living drugs, but more like they're the conventional drugs with predicted PK/PD, with predicted expansion and inevitable contraction, right? And so you can dose the patient, you will know that your product will undergo expansion and then will inevitably kind of subside and vanish. And it is much easier to kind of predict those and manage clinically, those therapies that have a defined lifespan. Now, for this modality to happen, first of all, you should engineer the cells to have this predicted behavior, right? But also it is very important that they are resistant to rejection, because if they induce rejection, then every subsequent reinfusion will not be as effective because the cells will be rejected right away. And so once we kind of establish this concept of a product that resists rejection, either through ADR or maybe through the stealth mechanisms, then we can approach this modality of having kind of cell products with predictable pharmacokinetics post-infusion, which I think this would be the future of all cell therapies. And this will allow you to stop them once the tumor melts, completely kind of dissipates, or if a tumor mutates and loses the target antigen, you just inject a different product, right? That will go after another antigen.
Got it. So in terms of the rejection that you're talking about, getting rid of rejection definitely seems the first step of making something like this work in the first place. And a lot of people think that, okay, so we need to have HLA matching to make sure that these cells are not rejected. And there are those who suggest that we can have these banked HLA homozygous cell lines, and we have maybe about 30 to 80 treatments depending on the population, and you choose one and you give it to the patient. How feasible do you think something like that is? And I also wanted to ask you about HLA matching in general, because you can't match for everything. That's way too hard. And how do you determine, okay, we should match for these 4 genes and everything else is fine, or maybe just 2 genes is okay? Or how does that sort of work?
Yeah, this is a great question. It's a little bit outside my expertise. I think the best people to answer this question would be transplant physicians, right, who deal with HLA matching all the time. But it's a very interesting approach, and I think it has a lot of merit. you can cover, I think, about 90% of the population with just a handful, or maybe 5 to 7. I don't remember the exact number of HLA-A alleles, right? And if you think about less polymorphic alleles like HLA-C, that's probably even better. So you have fewer products that would cover reasonable, number of people. But I think that the big question, as you alluded to, whether matching on 1 or 2 alleles will be enough to completely suppress NK cell activity or T cell activity, right? Because you still have some HLAs, and you still have some, potentially immunogenic proteins inside, or alleles that are not present. And so these cells will not be completely non-immunogenic. They will have reduced immunogenicity. And whether this immunogenicity will be reduced enough to evade the immune response will depend on many factors. First off, how well the HLA is matched, right? The second is, it depends on the particular patient's immune system. Like, there are a lot of patients with suppressed immune systems due to lymphotherapies, chemotherapies, and lymphodepleting drugs, right? And their immune system may not mount a sufficient, reaction against those products. And so it will be, I think, a little bit more heterogeneous in patients, and will work in some and work less so in others. But at some point, it's a numbers game. If you have if you have enough of your therapeutic cells that produce that activity, you don't need maybe to have it completely invisible to the immune system, because there are other mechanisms that may limit the rejection, such as development of microtolerance, and those may kick in. And so it's just that it's difficult for me to say whether it will work or not, just because there are mechanisms that we probably still don't know how they work in the clinic and what their importance is. And I think it is critical that we try all these approaches in patients, see which one works and which one doesn't, and then maybe combine different approaches that have good safety profile in order to kind of create the ultimate off-the-shelf defense strategy.
Got it. So as we sort of slow things down and end here, I want to ask you what you think is next for the field of off-the-shelf CAR T-cell therapy. Obviously, there's a lot of stuff that's going on. You're doing clinical trials that are very important, and we'll see what happens with that. So where do you think this field as a whole is going, and what do you think some of the obstacles you and other people will have to face going forward to create these allogeneic stem cell therapies, or just cell therapies?
Right. So I think the two main problems, the two main directions, I should say, right now for the field are: one is, what is the optimal cell type that exists, right? Right. So there are many options for a cell platform for the off-the-shelf therapies. So you can use NK cells, you can use gamma delta T cells, you can use MATE cells, you can use iPSC-derived T cells, you can use donor-derived alpha beta cells that are edited, right? What cell type produces good enough anti-tumor activity, has good safety profile, and has desirable PK/PD characteristics? And the answer may not be just one. It may depend on the tumor type. Some diseases, like acute myeloid leukemia, may be more susceptible to, let's say, gamma delta cells or NK cells that have other ways to target myeloid cells through innate receptors. And then alpha beta cells or iPSC-derived effectors may be suitable for vast majority, just because they have alpha beta cells have proven track record in the field, and iPSCs are very pliable. You can engineer them inside out, right? You can make them alpha betas, you can make them gamma deltas. Right. So that's a very interesting approach. So that's one direction, and the other one is actually answering the question of persistence. How do we ensure sufficient persistence of off-the-shelf cells to not make it very toxic to the patient, to kind of move away from lymphodepleting therapies upfront, or carpet bombing with CAMPF shortly before, or kind of to get rid of some unwanted effects that come arise from immune suppression in these patients that currently is required to ensure persistence of the products. But ultimately answering the question how much persistence is needed, and then engineering T cells in a way that they have predictable behavior. And so that's the thing, that the next kind of 2 steps for the field. And ultimately kind of really engineering cell therapies, not maybe for cancer, but also for other grave diseases, such as autoimmune diseases or neurodegenerative diseases, transplant rejection, all this stuff. So that's a fascinating field. We're living in a sci-fi time right now without the fi part.
Right. And that's so exciting. How easy is it to manufacture these products in a GMP condition, a good manufacturing practice condition? Because you can have the therapies and they can work really well, but in terms of making this more streamlined for patients, is that a big hurdle, or do you think that's just something we have to just perfect along the way?
Well, I think GMP, we've learned a lot in the past few years, and in terms of GMP manufacturing of autologous products, and we have to sometimes adapt manufacturing for specific patients just because they have difficult kind of cases. But the thing about GMP manufacturing of off-the-shelf cells is you don't have to manufacture them as frequently. Like you scale it up once, right, and then you freeze a whole bunch of vials, and then you test them and make sure that they behave, they produce desired activity in preclinical models, and then you do a release test. So it becomes a question of scaling up rather than kind of solving specific problems. And so I think GMP manufacturing will not be a big hurdle, especially when we figure out how to grow iPSC-derived cells and kind of establish master cell banks that they can then be differentiated continuously through working cell banks and other approaches. And then alternative approaches that deal with the generation of off-the-shelf cells from healthy donors. It's a bit, probably there's more variability in there, but all in all these methods have been established really well. So like I said, it's more finding solutions to scale them up and ensure good potency as you grow the cells in large quantities to avoid excessive differentiation due to prolonged culture and things like that. So I'm optimistic in this regard.
That's great. And it's great to hear that there's so much going on and there's still so much to be solved in this field. I guess the questions keep coming as you keep exploring more. So thanks so much for your time. And I just wanted to end on a brief question for those who want to pursue bioengineering, want to pursue cell engineering. What advice do you have for students who are either in high school or who are in undergrad who want to pursue this field?
Well, it's an interesting question. My advice would be simple: try to expose yourself to as many things as you can. There's a lot of fascinating stuff happening in this field. There's a lot of groups that are exploring, wonderful approaches that may be completely, the next big thing. And there's so much to learn before you kind of embark on a specific project. So when you're just starting out, I would advise you to try different things, to see what are the great new ways to edit out genes, right? What are the great new ways to engineer cells? How we can engineer synthetic biology circuits inside of cells, how we can differentiate induced pluripotent stem cells into what we want. There's so many things if I were a high school student right now, I'd be like a kid in a candy store, really not knowing where to start. So find your passion. There's so much to choose from, and once you find it, just dive deep and follow your passion.
Sounds
It's
great.
fascinating. So.
Yeah, and thanks so much for the interview. I really appreciate it.
Yeah, it's been a pleasure. Thanks, Sid.
All right, and that's the wrap. Thank you to Dr. Mamonkin for taking the time to speak with me. You can read more about his work on my blog, Brooklyn Musings, and I'll see you next time on Entangled Strands.